Hello, everyone. So you're welcome to lesson number 4, gene regulation and mutation. So how do prokaryotes and eukaryotes, how do they regulate the genes? Gene regulation is a must because you have to control the rate at which you produce several specific type of proteins. So if the genes aren't regulated, that could cause a lot of diseases, waste of resources, and cancer. So this is why they need to be a regulation in gene that produce several proteins or the processes of producing a specific protein. So among the things we're going to talk about is gene regulation. We're going to mention operon, mutation, and mutagen. Prokaryotic organisms like bacteria, they does not have a membrane-bound organelle. This is one of the main qualities they have. And they also, the DNA is organized in the chromosomes. They do not have a membrane-bound organelles, and they don't have a nucleus. So the genetic material is organized in the nucleus, in the chromosomes. So gene regulation is the ability of an organism to control which genes are transcribed in response to the environmental changes. If a person gets to a new environment and he wants to adapt to the environment, so there will be some gene regulation that would happen to make him produce some proteins that can make him cope with his environment. So the ability of an organism to be able to control which genes are transcribed to be able to cope with the environment is called gene regulation. An operon is a section of the DNA that contains the genes for the proteins needed for specific metabolic activity. We have different types of operons, and the operons are a section of the DNA that actually has those genes responsible for the production of the protein needed for specific activity. What should be contained in the operon? Among the things that should be contained in the operon is this operator, which is a one that turns on and off the process. We should have a promoter. The promoter is where the RNA polymerase binds to. We need to have a regulatory gene, and we need to have the genes that code for the main proteins we want to print. We're going to take as a case study the TRP operon. The TRP operon is called the tryptophan operon. Tryptophan synthesis happens in 5 steps, and all of these 5 steps are controlled by the TRP operon. Every step is being controlled by a gene, a specific gene. So the TRP operon is a repressive operon because it usually repress or turn off. The meaning is the TRP operon usually do the normal thing. It usually carry out its function of transcription, but it could be repressed. It could be turned off if there is no need to make tryptophan. Let's take a look at an example of tryptophan operon. So this is a DNA, and upstream the DNA, you have a repressor gene. You have a promoter region, and you have the genes responsible to print the proteins which you need. So the DNA for the repressor gene will be transcribed into mRNA, and the mRNA has a 5' to 3' will be translated into a repressor protein. So the repressor protein is inactive here because the process of transcription has to proceed. So once the RNA polymerase sticks to the promoter region, it transcribes toward the gene to produce a code for production of tryptophan enzymes. So the tryptophan enzymes then can do all the activities it has to do. So at a point in time when the level of the tryptophan is a lot in the system and you don't need to make more of it, so what happens is when the DNA is being transcribed into mRNA that usually produce a protein which is needed for the process, the repressor protein. So the tryptophan would stick with the repressor protein, and it's going to inactivate, it's going to make it to move and stay in the position of the RNA polymerase. So the RNA polymerase would not be able to proceed with the process of the transcription. So what happens here is the tryptophan operon then gets turns off because what? Because there is a repressor sticking to the position of the RNA polymerase. The second we want to look into is a lac operon. A lac operon is found in E. coli, Escherichia coli. So this operon is very useful when the lactose is present. The E. coli can synthesize an enzyme to use it as an energy source. But the lac operon is an inducible operant because an inducer binds to the repressor and inactivates it. So it allows the transcription to be turned on. So let's see the process in real life. This is a DNA. Upstream of the DNA, you have a repressor gene. Then you have the promoter region here. Then you have the gene to be transcribed in the front. But the problem here is because it's lac operon, it's automatically having the promoter sticking to having the repressor protein sticking to the promoter region. So this will be stopping the RNA polymerase from proceeding with the transcription process. So this is the mRNA for the repressor gene. This is a protein for the repressor gene is active. So it goes and stick to the position of the RNA polymerase, and the transcription process will not be able to move on. So the repressor prevents the transcription. Now, what happens is when there is when you have lactose in the body, so the lactose usually come with an inducer, so called allolactose. So the allolactose, when the repressor gene is being transcribed to mRNA, then it gets translated to repressor gene. Then the inducer gets into the system because you consume food that has lactose. So the allolactose, which is in the food, will stick with the repressor. So this will prevent the repressor from being able, because it's going to inactivate it. It's going to prevent the repressor from going to bind to the promoter region, which usually stop the RNA polymerase from doing its function. So what happens is when the allolactose inducer, when it sticks to the repressor protein, so the repressor protein could not be activated to go and stick to the position of the RNA polymerase in the promoter. So the RNA polymerase now is in position of the promoter, and it can run the process of production of all the genes which are responsible for the production of enzyme lactose that can break down lactose. So all of these enzymes are different enzymes for lactose digestion. Now, controlling transcription. Transcription factors ensure that a gene is used at the right time and that proteins are made in the right amount. There are two main types of transcription factors: complexes that guide the binding of the RNA polymerase to the promoter, because guiding the binding of RNA polymerase to the promoter is what starts the process of transcription. If anything stops the RNA polymerase from binding to the promoter, or the promoter is being blocked, there would be no transcription. Regulatory proteins that help control the rate of transcription. So there are some regulatory proteins that can help to control the rate at which the transcription is happening. The complex structure of eukaryotic DNA also regulates the transcription process. Eukaryotic organisms regulate their gene transcription process or translation by using OX genes. OX genes is called homeobox gene. So this gene regulates regulation is very crucial during the developmental stage of the kid's formation or young formation or reproduction or the cell differentiation. The cell differentiation is a process which a stem cell decides to become different types of cells. One group of genes can control cell differentiation. It's called the homeobox gene group. So the OX genes are transcribed at specific times in specific places on the genome. The OX genes help to control which part of the body would develop a given body location. So it tells which part is going to become the head, which is going to become the chest, the stomach, and the likes. So the OX genes are responsible for general body pattern of most animals, including humans. Notice that the order of the gene is the same as the order of the body section in the genes. You got them here. So all of these genes, for example, the green one is responsible for this mouth region. The blue one is responsible for the mandible region. The ash one is responsible for these and like that. So this is the way the genes function. So every gene is responsible for every body part. RNA interference is another method by which the eukaryotic organisms can regulate the genes. RNA interference is called RNAi, can stop the mRNA from translating its message to produce proteins. Single-stranded small interferon RNA, and protein complexes bind to the mRNA and they stop the process of translation. So this is a protein complex here. This is a single-stranded small interferon RNA. So they're going to bind this to the position where the transfer RNA is supposed to bring the amino acid to translate it to get the codon translated to amino acid. But because this complex is blocking it, the process cannot proceed. Now let's talk about mutation quickly. Mutation is a permanent change that occurs in a cell's DNA. So when there is a change that is not reversible, it's called mutation. We have two main types of mutation. We have point mutation and we have a frame shift mutation. The point mutation can be described in two ways: missense substitution and nonsense mutation. In the missense substitution, the DNA codes for a wrong amino acid because there was one nucleotide which is not correct. So by the nonsense, the codon for amino acid becomes a stop codon. So because of the removal or replacement of one single nucleotide base, so it led to the formation of a stop codon. So the process of the transcription or translation stops. Another one is insertion or deletion. The insertion or the deletion can lead to a frame to shift. So the frame could shift forward if there is, let's see the example in the next page. Let's say this is a normal normal DNA sequence. It's used in a sentence. So when the missense mutation happens, it would remove one nucleotide base, G, and replace it with a Z. So this is the only thing that happens, and it could lead to achondroplasia, where the arms and the legs become short. So it has the missense. So one nucleotide got replaced or got changed. In the nonsense, the same thing happened. The F becomes the R, but this RAT is a stop codon. So the transcription or translation process could not continue again. So because the transcription stops, so that's why you see the other ones are not printed. So this can cause muscular dystrophy. But as for deletion and insertion, you would see here the E in the B got deleted and the B got moved backward. So this is why you have the B here and IG move backward and everything move backward. And you will see the AT was the last remaining. So this can cause cystic fibrosis. The meaning is because there was one nucleotide base being deleted. But when you insert, you actually inserted an additional nucleotide base into it. For example, you inserted that before the F in fact. So you had SFA. It's going to move the frame forward. It's going to shift the frame forward. So that's called insertion. That could lead to Crohn's disease. There could be sometimes also be duplication, whereby some codon are printed multiple times. So if it's printed 2 times, it's duplication. If it's printed multiple times, it can lead to expanding mutation. So sometimes when there is mutation, small changes in the DNA code can cause genetic disorder. A change in one amino acid can change the sequence of the protein enough to affect both the folding and the stability of the protein. Sickle cell disease is an example of a disorder caused by a single point mutation. Now, let's take a look at what actually causes mutation. Mutation can occur spontaneously. Spontaneous mutation is when it seems not to be any reason, or it could just happen from nowhere without being expected. So DNA polymerase can attach to the wrong nucleotide. So when a DNA polymerase attaches to the wrong nucleotide, so this could lead to mutation. It's a very rare case, but it happens. We could have certain chemicals and radiation, which is called mutagens. So these mutagens can damage the DNA. Chemicals can cause mispairing of base pairs or themselves substitute for base pairs. So we can have some causing a mispair, for example, causing A to pair with G, for example, and causing C to pair with U. So this is called mispairing. High energy radiation can also eject electrons from atoms within the DNA molecule, leaving behind unstable radicals. So body cells versus the stem sex cells mutation. The body cells are called the somatic cells. When there is mutation in the somatic cells, the mutation could not be passed on to the next generation. So it's not something that could be inherited. But the mutation that happens in the sex cells, they can be passed on to the organism's offsprings. And this will be presented in every cell of the organism, of the offspring they have. So this is an example of a mutation that can be passed on to the offsprings. So we're going to stop here. So I wish you all the best. See you again next time.